The rapid advance of science presents us with the hope of eventual cures for terrible diseases. And with profound moral and ethical dilemmas. In the complex debate over embryonic stem cell research, we must remember that real human lives are involved. Both the lives of those with diseases that might find cures from this research and the lives of the embryos that will be destroyed in the process.
Embryonic stem cells.
Embryonic stem cell lines.
Embryonic stem cell.
Embryonic stem cells.
Or embryonic stem cell research.
Embryonic stem cells are derived from blastocysts, embryos that are about a week old. At this stage, the blastocyst has about 100 cells. Human blastocysts like this have been donated to research from in vitro fertilization clinics.
Once the egg is fertilized, it divides into a ball of cells. After 4 to 5 days, scientists remove the cells from the inner cell mass region.
These cells have the potential to develop into any type of cell in the body. Once the cells are removed, they are placed on a culture plate with nutrients and growth factors. The blastocyst is destroyed in this process. An embryonic cell line is established when these cells multiply and divide. Under the right conditions, these cell lines can be maintained indefinitely.
They are then transferred to a laboratory dish where they continue to grow and divide. By adding different growth factors, it's possible to change these embryonic stem cells into different cell types, such as nerve, bone, muscle, and pancreatic.
The latent potential of human embryonic stem cells is one of the most exciting fields of research in medicine today. Scientists believe these minuscule cells hold the promise of better treatment for some of the world's most dreadful and debilitating diseases. Hadassah is considered as a pioneer and a world-leading research center in this field. Stem cells are formed inside an embryo 5 or 6 days after fertilization. These cells can produce themselves indefinitely and can develop the characteristics of any type of human cell. Hadassah's pioneering research in this field continues to reap international acclaim as our physician scientists continue their efforts to develop stem cell treatments to rejuvenate disease-ravaged systems. Once this capability is achieved, medicine will have inexhaustible pools of cells that will help cure all kinds of degenerative diseases.
Is human life inherently valuable? Does it deserve protection simply because it is human life? Over the millennia, the Christian position on the issue has been clear. Because human beings are created in the image of God, they have inherent value. But in recent decades, the issue has become confused for many. This issue becomes even more confusing when it gets down to the microscopic and embryonic level.
Human embryos are human life.
Is a person. That's a life. That's a human life. Yes, it is.
The issue of embryonic stem cell research has been a political hot potato, with debates about federal funding having come before the United States Congress. But what exactly is embryonic stem cell research? And why do those who hold the pro-life position oppose it? Dr. David Prentice, Family Research Council.
Given the correct signal, you can get that cell to form any of a number of specialized tissues in the body: nerve, brain, heart, liver, kidney.
Diseases that will be approached and cured most likely are neurodegenerative diseases like Parkinson's, Alzheimer's, spinal cord injuries, so those kind of paralysis kind of things. We think we can attack those pretty easily with stem cells and robustly. We're learning about cancer in ways that we never thought. Maybe the best way to treat a tumor is not necessarily to kill it, but to change it. What if you could turn it into something less harmful? And that's kind of a strategy that our lab has adopted.
The undifferentiated cells, called stem cells, are found in embryos up to 14 days old with the ability to become any one of the 220 cell types in the human body.
Embryonic stem cells are taken from an embryo that's just a few days old.
The real tricky part here is that because one of the sources of stem cells is the human embryo, it is still impossible to tease out those cells from the early embryo without not just harming the embryo, but actually killing the embryo. So the question of embryonic stem cell research is obviously connected with the question of manipulation and moreover exploitation of embryos.
Dr. Pia De Soleni, an ethicist and moral theologian, is a specialist on life issues.
When those stem cells are taken from that embryo, the embryo therefore dies. And the embryo is a human in its very beginning stages of life.
There are also practical problems with using embryonic stem cells. Dr. Kelly Hollowell is the founder and director of Science Ministries Incorporated. She is a bioethics expert who holds a doctorate in molecular and cellular pharmacology.
Use of embryonic stem cells has major problems associated with them. The two biggest problems are immune rejection of the stem cells, and the second is growth of tumors.
The major ethical controversy has surrounded the use of embryonic stem cells, and more specifically, it's the controversy surrounding the use of embryos to obtain the embryonic stem cells. So a lot of the controversy has to do with how we get the stem cells that are found in embryos.
When the people go get IVF, in vitro fertilization, and they test tube baby kind of thing, they grade the embryos that they make, and the best ones go into the top 5% go into the woman's uterus, and only maybe 1 or 2 makes a baby. Inevitably, there's about 150 embryos that don't get used. Those embryos get frozen in liquid nitrogen, but eventually that liquid nitrogen runs out. If you don't pay your bill or whatever, they get thrown away, so they're destroyed one way or the other. We just look at it as like, at least it's going to a good cause.